Welcome to another segment of Engaging the Big Questions. I am Doris Jones from the Academy of Liberal Arts Integrative Learning Initiative, and today we are honored to have with us Dr. Anwar Abdelnasser, who is an assistant professor in the Institute of Global Health and Human Ecology. He is also the principal investigator for the COVID-19 grant project, which is entitled ACE2 and TMPRSS2, the Achilles heel of the SARS-coronavirus to SARS-CoV-2. What is fascinating about your co-principal investigators is that this is an interdisciplinary team. You have several of your colleagues from the Global Health and Human Ecology Institute, as well as a colleague from the Department of Mechanical Engineering.
Yes.
What allows for this interdisciplinarity to bring about strengthening the research that you're performing?
That's a really nice question. Let's start by the first co-PI, Dr. El-Fawad, who is an expert in global public health, and given his expertise in different areas related to health, mainly pharmaceutical sciences, biomedical sciences, and biomedical research. His expertise was really wanted in this project. Dr. Salama, he's an expert in the genetics area, and his work on the Egyptian reference genome was the laying foundation from which we were able to start our work on this project. Dr. Ahmed Mustafa is a fantastic bioinformatician and biostatistician, one of a few in Egypt who are able to do such neat work related to bioinformatics and biostatistics. And last but not least, Dr. Mustafa Yusuf, he's part of the Department of Mechanical Engineering, but he has also fantastic work related to molecular dynamics simulations and interaction of different biological components with each other through molecular dynamics using computational forces that would help us better understand the interactions of different proteins, different nucleic acids, part of the cellular systems, together in one great way to visualize how the interactions would take place.
Certainly. And again, a collaborative team working together, interdisciplinarity, all for the purpose of advancing this wonderful research project. Which brings me to another question. How did the idea evolve to perform this research project?
The idea evolved once we, if you recall, we were all affected by the pandemic
Yes.
early 2020, although it started spreading out from China by the late 2019, and hence the name COVID-19. But at the beginning of 2020, we started suffering the impact of, at the time, the WHO started calling it a pandemic. Part of this pandemic was an imposed lockdown on different levels, including the AUC. At the time, the provost and the associate provost office came up with a fantastic initiative to support COVID-19 research. Given that I'm a pharmacologist / toxicologist by training, I try to figure out a way if we can do some wet lab research. Unfortunately, we have very limited capacity building to do virology research in Egypt. And at the time, I think there was almost close to none labs that were able to host such virology research. So we tried to find out a different approach by which we can conduct similar work, but relying on biostatistics and bioinformatics, and taking into consideration that the sequence of the virus has been already identified. And based on these facts, we can easily start examining the interaction of the virus with the components of the cell, such as the ACE2 and CAMPRES2 that are part of the grant title, and how would this interaction be in different populations, different ages, different sexes, between males and females, people at different age, and across different populations, whether it's Africans, whether it's Asians, Caucasians, and most recently we were able also to withdraw that to the Egyptian population based on the study was done by Dr. Salama and his colleagues in Lübeck in Germany, for Salah Ibrahim and the others. So based on that, we were able to come up with a fresh idea, trying to examine this interaction and trying also to find a way to intervene between the interaction of the virus itself with the components of the cellular system that would permit the virus to enter and fuse into the cell. ACE2 is one of them. TMPRSS2 and furin are also other components that are important for the virus for its entry. And this is the first step in the life cycle of the virus, after which it will be able to continue its production and manufacturing by hijacking the cellular system and producing more and more virions or virus particles that will be able to infect other cells.
And speaking of which, that explanation is certainly vital to understanding the virus itself. What are you and your colleagues, how are you addressing or how are you thinking about the current Delta variant that has become quite prominent around the globe, especially in the United States? I'm learning that hospitalizations are up by 500%, primarily because of the Delta variant. How is this mutation of the virus impacting your research?
That's a very excellent question, fantastic one, in fact. It does affect our research because it's very hard to predict the mutations of the virus. And when would these occur? So each time or every few weeks or months, you find that a new variant has been identified. And it's very difficult to have a system to predict these variants. Nonetheless, the interactions with the components of the cellular system that we have included as part of our proposal would remain the same in terms of the proteins we have. So in this part, we would have the sequence of the virus with the variants included would be our variable that we will need to try to have a simulation to find out how would this behave in the different populations again. And this gives a continuity to the COVID-19 research, which was initially thought that it wouldn't last more than a year or a year and a half at the most. But given the number of variants that are emerging since then, I don't think that this is going to be something that can end soon in terms of research or in terms of vaccine developments. It would continue to be a very hot area for the next few years at least, especially that with the new vaccine strategies, people thought that it would be the end of the virus, but nonetheless the virus strategy seems to be also smart by developing different variants trying to escape the immune system as much as it can.
Fantastic. So the very mapping of the virus itself can in some ways help to circumvent or even perhaps, I gather, lessen the concerns, overwhelming public concerns about the replication or the mutating of the virus itself, as long as there is a clear understanding of the variants, the sequence.
Yes.
Excellent. Very nice. Which brings me to yet another question. So since the research project has begun, what have you learned about COVID-19?
We have learned a lot from our research and from other labs around the globe that are conducting COVID-19 research. One of our first lessons that we came up with is that in the different populations, you can have the genetic determinants will determine the severity of the case with the interaction of the virus with the host cells. Given that also we looked at the differences between males and females, and we learned that in males, the severity of the cases are higher than females, given that one of the proteins, which is TMPRSS2, is more elevated in males compared to females, and this is naturally occurring because of the sex hormones. Also, in different ages, children might not develop severe cases compared to adults, also due to the differences in the expression and the variants of the proteins inside our host cells that would also dictate the severity of the cases. Doesn't seem to be occurring in children. Nonetheless, we also found out as part or as a laying foundation of what we have done so far in this research, that there are long-term complications that are associated with the COVID-19, even for those who were treated and they are no longer hosts of the virus. One of the most important outcomes of that would be some neurodegenerative diseases and neurological disorders. And we have already started developing some neat research within the Institute, myself, Dr. Salama, and Dr. Ilfawal, trying to figure out what could be the biomarkers, or how can we identify if a person who has been infected with COVID-19 would develop some of these neurological disorders. And this is also in collaboration with Mansoura University, by which we are trying to obtain some clinical samples and try to analyze them here at the AUC.
And this fits very nicely into the next question. These are some of your key findings in the research, that there are higher incidences among males, that you're finding that children are not as susceptible to the virus, and that certainly even in cases where persons who have been infected by the virus, that there may be long-term implications, primarily neurological in nature,
Yes.
and the implications that that has. Are there other findings that your research has yielded?
We are currently finalizing a study in which we developed a vaccine that would be a pan-variant vaccine covering the different variants of the virus. It's coming from a different school compared to the current vaccines in the market, whether they are attenuated or killed virus particles, or mRNA-based or DNA-based. The vaccine that we came up with, its design, is based on polypeptides or small amino acid sequences. And they are connected with each other through a small peptide connection, so that we can, once it's inside the cell, it would be broken down into smaller pieces, and each one of these pieces will be working out to fine-tune the immune system as kind of synchronous performance to come up with an antibody that would be able to neutralize the virus.
As you can imagine, we are wishing you and your colleagues much success, because the development of such a vaccine will certainly be needed. This research is outstanding, which brings me yet to another question. So, why did this research need to be performed, given its uniqueness? And I must also add the speed with which you and your colleagues are able to move through with developing research, publishing. During our initial conversation, you shared with me several articles that you and your colleagues have published that brings up to date your findings, considering that this grant was disseminated just a little over a year ago. And I'm listening to all of the successes that you and your colleagues have been able to produce. So why did this research need to be performed?
Honestly, the initial way of thinking when we wanted to do this research, we figured out that the number of COVID-19 cases in Egypt were much lower than what we were expecting based on the numbers we hear from other countries. And at the time, we wanted to figure out if there could be an explanation based on genetics that could explain to us that we don't have the number of severe cases similar to the other countries. And that was the initial trigger to do this research. And this is how we came up to start comparing the genetic sequences of the Egyptian population to different other populations. And I think it has in part explained the lower number of cases in Egypt compared to other populations. This is just looking at the genetic predisposing factors, but we need also to look into the environmental factors, the adherence to the different aspects, and hence the other publications that you were referring to, into which we were trying to examine the environmental and occupational risk factors that could be in part responsible for the spreadability and the severity of the COVID-19 cases.
So again, considering that there was initially a lower spread of COVID-19 in Egypt, which certainly precipitated an interest among you and your colleagues to examine, as you indicated a moment ago, what are the factors contributing to the lower spread, which is good, of course, from a public health perspective, which brings me to yet another question.
Yes.
So what then are the challenges, if any, you have encountered, and how are you overcoming them?
One of the challenges we had in our research, as I mentioned before, is having a wet lab research.
Right.
And testing our vaccine in-house would be a challenge because we need to have a certain mouse model, certain cell culture model, or wet lab research model. And it could be basically very, very difficult to do so, given that you would need to have a production of the virus. So this might be a spot where a virus spread can be initiated, so it needs to be conducted carefully. The biosafety level that is needed for such work is biosafety level 3 and higher, and we do not currently have such facilities that are part of the Egyptian universities. The latest was an initiative with Italy in Alexandria University, but I think the lab is still under construction, so it's not yet operative. So for that, although it was a challenge for us and something that we see is not going to be an easy task to do in Egypt, I tried expanding the type of collaboration we can have from outside Egypt. And I tried communicating with Professor Andrea in Italy, who is currently also working on the same topic, but the lab from which they are trying to test different drugs and different vaccines is located in Brazil. So we communicated with each other. He visited me here at the AUC a few months ago, and we are currently trying to figure out a way by which we can test the developed vaccines or the developed vaccine that we came up with in Brazil or in Italy, if he would be able to support this initiative.
And again, that speaks to the beauty of the collaborative nature. This is an international initiative.
Yes.
In which certainly I would venture to think that you will be able to find answers to circumvent the current challenges. This is all very good. Okay, so what was the first step you made in this project?
The first step was trying to figure out the environmental and occupational risk factors that could be related to the spreadability and the severity of cases, whether it's in Egypt or outside. And at that time, I was teaching the course Environmental and Occupational Health within the Institute of Global Health and Human Ecology, part of the Master of Public Health program. And at the time, the students were really interested in the topic, and I gave them the initial idea, and they have contributed significantly into doing the literature review, coming up with ideas and covering the topic significantly. And we were able to publish this study early on October last year. And so far, I think it has exceeded 6,000 views on its publishing group website. And since then, things started developing even much faster with the comparison of the sequences, the bioinformatics related to the different alleles of ACE2, TMPRSS2, and purine. And we came up with our second study, which was published I think last month. It got published again recently. And then we came up with a third study related to the downside, because we needed to pave the way to our study about the vaccine.
Yes.
So we needed also to evaluate the current vaccines, the mRNA-based vaccines, and see what are the downsides of these vaccines and the way for their improvement. And currently, this study is under review. We received the comments last week that we are required to do some minor revisions, typographical edits, but the editors of the journal Vaccines are interested in the article as is.
Congratulations.
Thank you very much. And our fourth and last study would be the vaccine study, the development of the vaccine, which we have worked on really hard, and we came up with this neat study. But I'm still waiting to hear back from the Technology Transfer Office, because I think it's a very good idea to patent this vaccine before we can publish its content.
Absolutely. I think another commendable part is, in addition to the interdisciplinarity of the project, certainly the international collaboration that is involved. Our own AUC students are actively participating in the research project. You mentioned their engagement with the literature review, understanding the methodology, understanding more about the complications of the replication and mutation of the variants of the virus. It's so very important, as you know, as a researcher and scientist, that students do have an opportunity to work alongside someone like yourself to understand the process. So this is outstanding. My final question, what would you say to your supporters who have funded this work?
Frankly, I don't know the right words to express my gratitude to the Provost Office and the Associate Provost for funding this research. But I can tell you that without this support, we wouldn't have been able to publish such fantastic work. We wouldn't have been able to conduct this work to start with. And also, I need to mention that not a single publication that came out from this work didn't have any student, or did have an AUC student on it. So all of the publications that are coming out, it either has one or more students from AUC and at least a couple of co-PIs who participated in the study. And I think this funding, although it might look a little bit smaller on scale in terms of monetary value, but it gave us a great support in the time where we had very little support from outside AUC. And I think it served the purpose of funding by initiating new ideas for future funding, which I have applied for, and I'm still waiting for the decisions for some of these grants. And I think it worked out beautiful in terms of supporting us all in terms of research, students' engagement, collaboration, you name it. So I'm really grateful for this support.
Dr. Abdelnasser, we are so very grateful to you. And as you can imagine, given the successes that you have been able to achieve with you and your colleagues, we're wishing you much continued success and that for the grants that you have recently applied for, that you will be able to receive funding to continue and carry on this very, very important work, not only for the citizens here in Egypt, for our immediate institution, the American University in Cairo, but also for the rest of the world. Mabruk.
Thank you very much. Allah barik feek. Thank you very much.
Thank you.